going to get started. I hope you can all hear me, and if you want to see me and see the screen. So before I begin on formal matters, the way it's going to work, as with the other ones, is that I'll talk for an hour or just under, and you can use the Q&A section of Zoom to actually provide some questions, which I'll then go through at the end, and so you can sort of hopefully get some of my attention after this is over. Okay, so welcome. This is an overview of bioinformatics for biologists. It's not for people who already know lots about bioinformatics, really. So bioinformatics is an enormous subject, and I'm not going to be able to cover, even 1/10 of it. So all I'm going to do is just give you a brief overview of what kind of things you can do, and perhaps try and be helpful by giving you tips on how to get started on particular aspects of bioinformatics. Now, in the olden days, particularly, people used to think bioinformatics was just sort of aligning sequences, and yeah, a lot of it is that, of course. But really, I like to think of it as the intersection between computing and biology, and a lot of that also intersects with maths. And so, if you want to do serious bioinformatics, of course, you have to get into a bit of maths, and I will be putting some helpful pointers to do with statistics. Which actually, I get quite a lot of questions about that here at the LMB. So let's get started on this. Oh, some people out there might like this cartoon. Epigenetics is just extra formatting. There we go. That's a bit rude, really. But okay, so when we're talking about bioinformatics, you can think about the information flow through the central dogma, as it's called. But of course, there are many more types of molecules we're also dealing with these days. There are many different organisms that we're dealing with. And how do you tell about function? It's not just from sequence. You've got to think about the structures that have been determined, all the interactions. It's a vast, vast area. And even these days, of course, we're talking about how even the structure of a chromosome can affect the expression and the folding, and all the rest of it. Okay, so if you want to do bioinformatics, I guess the overarching thing is it depends on how committed you are and how much time you are. So I'm going to be sort of giving you a light touch intro about how to, how you can dabble as a biologist using mostly web-based stuff. So all the usual things to do with alignments and searching for homologs, and even actually protein folding these days. You can try online, and it's all tends to be publicly accessible. If you dabble, you can get a bit of help locally. In your institutions, there will be people like me who you could just come to for a bit of advice about what you should do, what not to do. Okay, and also these slides, you can use them. Later on, we'll try and make some version of them available, as well as the actual video itself, so you can have a look at some handy points to get started. But if you're going to get serious, you can become a computer person like me. And if you want to get started in that realm, I recommend, well, not only learning a bit of programming, and my recommendation there is Python, which is the easiest programming language to learn, but it is not a weak language. It's very powerful, but it's a good place to get started. And also, you're going to have to learn a little bit to do with the command line, so that's bash shells, a bit of Unixy type stuff. And I teach courses on computer programming and this sort of thing. And the notable thing is that those who get on best with it overall tend to be the people who really have a project in mind. They've got something they want to do. This might be something novel, or it could be just repeating something somebody else has done. So if you want to really get into the deep programming side of things, have something in mind, have some data you can work with. And also, eventually, you're going to have to learn a little bit of stats, or maybe even probability as well. Okay, so overall, this is all about commitment. Okay, on to some databases. So yeah, you want some data, you can collect your own, but there's an enormous amount in the public databases these days. Of course, everything changed as soon as we started sequencing whole genomes, doing high throughput experiments and whatnot. So this guide I'm going to give you here, it's about big data. It is constantly changing, of course, new stuff comes along and it can't be exhaustive. And it's just, what I take notice of today. But again, hopefully some handy start points. There's a relatively recent review here of lots of molecular biology databases, a collection in NAR. So you can look there if you want a more exhaustive reference. Now, one of the things I do when I need to know what kind of databases there are available is I just go to hub websites. So typically, I want to know, for example, what databases of proteomic information are there. For example, if I look in Uniprot, you can see cross-links to lots of databases. So these are the ones that it reckons are good enough to actually provide some information for the people who visit the site. Verification. I'd have had no idea where to start. I go in and see what, effectively, this is what Uniprot is recommending to me. So use hub websites like Uniprot or Ensemble, etc., and you'll find in there that you'll have cross-links, and these will give you hints about all the kinds of databases. And of course, I don't have time many of these. Okay, now so let's start thinking about nucleic acid, and the nucleic acids, like many databases, you can sort of think of them a bit split between those in Europe for the most part. So we've got NCBI databases, that's Wade's information, but of course, and there's Ensemble, which is a mostly European affair. And when you get genome sequence, the sequence, I mean that's not much help to a human being, reading it on screen. Of course, what you're interested in is all the annotations where the genes are, the export transcripts and proteins, etc., come out. So that's all the kind of data that's housed in these databases. So these are the two main ones if you want that kind of information. But one of the ones I tend to use quite often is UCSC genome browser as well, and it's just a bit more friendly, I find. So there's lots of handy stuff for you to download, chromosome sequences and whatnot. If you're interested in just specifically genes, well, then there's GenBank that has everything in there, and that's actually broken down into some subsections. If you just want stuff that is cDNA, there's this EST section for that. These genes, databases of genomes and genes, as you might expect, there's where to go. Now, something that's also, of course, DNA sequences are vectors, plasmids, all the rest of it, plasmids. And they have their own little database, UNIVEC, and that's really handy for finding contamination. Actually, if you include those in some of your alignments, sometimes you realize, there's, shall we say, it's not a natural sequence. So if you're interested in sort of screening out these sorts of things, depending on what you're doing, and then having a certain UNIVEC can be very handy. In fact, there's been a few genomes of certain animals. You realize they have done this sort of thing properly because you're actually getting vector contamination in what they think, trying to tell you is their genome sequence. Okay, so there's a lot of data there. You don't want to have to worry too much about computer programs. So use a genome browser. And there's many out there, particularly if you're interested in a specific model organism. But the three main ones I would recommend are the Ensemble, you may already be familiar with. There's a really pretty one that's been updated. That's the WashU browser. If you want sort of the graphics, that's a good one. And also my friend at the UCSC. This one, in a way, is a bit more sort of friendly and accessible. Ensemble is huge, if you like, and contains everything. So have a look, see which you prefer. And by genome browser, I mean this sort of thing here. You can hop along. It'll give you little sort of graphics representing where the genes are. And of course, these days, there's a huge amount of information, sometimes too much, that you can sort of lay underneath. For example, where are, the alignments from other genomes or species or whatnot. But you could superimpose epigenetic data on there, anything related that as a genome position, you can splat on there. A lot of these tend to be provided automatically, but things like the WashU browser, you can actually upload your own stuff and put that on too. So it doesn't have to be public. And it's all web-based, it's all accessible. One thing I really like at Ensembl, which I recommend people consider if they need genomic data, is to try this. So I've just put on this Biomart section. And what the Biomart section of Ensembl does is it allows you just to select the columns of data that you want from whatever section of their database. So for example, here I want to look at Castaneous mouse genes, but I don't want all fancy IDs and things. I just want to know ends and where the transcription start site is. This may be interesting in my research. So I've just selected the columns I'm interested in, and I can download it in lots of different file formats, including for spreadsheets and tab separated. Okay, so that's a really good way of just bulk downloading data in exactly the the column format that you want it. I find that very handy indeed. Okay, sort of moving on from the genome to beyond. If you're interested in expression databases or epigenomics, so epigenomics, I include epigenetics, so methylation, also anything on the genome like ChIP-seq, that would tell you, for example, where transcription factors are, where histone modifications are. Things like ATAC-seq, where the accessible regions of a chromatin is, and also, of course, RNA-seq. Then some big data that you can have a look at. And the main deposition these days for epigenomic and epigenetic data tends to be the GEO, which is run by the NCBI. It started out with microarrays, but it's just lots of high throughput sequencing data. So if you've got RNA-seq, ChIP-seq, etc., there's a lot in there. There's also a very good repository from ENCODE, and this was, of course, an organization where they had lots of labs distributing a lot of look at everything from chromatin structure, through the chromatin interaction, modification, and transcription as well. And the great thing about ENCODE is everything's very consistent. I was doing some studies of transcription factor binding recently, and I just went straight to ENCODE because they have everything in the same way, and they use the same formats, and it's all been checked, and it's reliable. Thinking beyond and toward transcription, there are transcription databases, and these tend to be most useful where the experimentation is consistent. So, of course, the amount of sensitivity you have in your experiment will depend on how you did the experiment. So it's a problem if you're looking, for example, in different tissue types, but the experiments have been done in different ways, and it makes it difficult to compare. So the best transcription database where it's done in a consistent and high quality way. There's Expression Atlas, the EBI, which kind of takes everything. BG is something that actually I've been working with people in the lab on. And this is quite a large tissue types. I think there's about sort of 80 to 100. And you can go in and you can actually look at transcription in a lot of tissues. And also, I've become aware of this thing called DepMap. Now, this is about cancer cell lines, and they have expression data in there. Perhaps more interestingly, DepMap includes measures of gene essentiality. So whilst you might not be interested in cancer, you can see they have sort of over 1,000 cell lines. They've done loads of expression and essentiality experimentality. I mean, they've knocked out practically every gene they can with CRISPR, and they look at viability of the cell lines. And this is really interesting information. Things might be in the same pathway or in the same complex, that sort of thing. It's very exciting and very good data if you want to actually get into some things. Okay, so going from transcription, there's lots of proteomics data, of course. The main repository is PRIDE, so you've obtained it. Of course, you can get other people's stuff too. There's big proteome databases beyond that, which are a bit more friendly. There's PaxDB. If you want to know the abundance of a protein, you can just go in and for different organisms very, very quickly sort of find that sort of thing out. Okay, so main sort of things above the genome there, and the main protein sequence databases, Europe split. There's the NCBI's offering, and there's the European offering, it's Uniprot. Here is much improved on what it used to be years ago. There used to be lots of different, protein sequence repositories, SwissProt and PIR. And what these two main sites have done is actually squeeze together lots of previous. They've formatted it in their own way, in a self-consistent manner. Although NCBI tends to have more in it, I prefer Uniprot on the whole for its web interfaces, and also it's got a sort of nice RESTful API, which means I can access things using IDs easily. So I'll be talking mostly about Uniprot, but you can do most of this at the NCBI if you wish. Just to draw your attention that there's different levels. A lot of Uniprot has not been checked, but a fair amount of it has, particularly in the model organisms. If you want the highest quality annotation, you want to trust something, then go for the SwissProt section. They kept that old name, and so SwissProt now is just a label for the curated prot annotations, and so you can really rely on people having checked through. For example, I was looking at membrane spans recently, and you can see that sequences, a lot of them are slightly wrong, signal sequences have been missed, etc. There's a lot of automated annotation helps, but that section has had people really look through it. Now, if you looked at the whole protein database, of course, it's vast. And yes, sometimes you do want to search and look into everything, but more often you just want a subset, and not all these databases allow you to check things and select particular sections. So frequently I'm looking, for example, only in the representative proteomes. You just want the best annotated version of each protein in each species, and also be selective about which species you're looking at. If you're only interested in yeasts, well then only look in yeasts sequence databases, and you can just click the taxonomy on in these things. Also, these subsets allow you to use redundancy reduced collections, which means there's it's only showing you sort of relevant information. It's also going to be quicker. However, there is a little bit of an issue actually if you just take proteomes, which is that the degree of completeness can vary. Fortunately, the UNIT website actually shows you how complete different proteomes are. So, for example, in this Drosophila species, there's a bit missing, whereas in the regular species, it seems all fine. Although the genome itself will have some duplication in there. But you can look basically at the quality of the proteome, and you use this information. For example, if you can't find something, well, actually, is the proteome complete? Not be. So yeah, these are redundant databases I've mentioned. Uniparc just has sucked everything in and grouped it. Uniref, in a way, is a bit more sort of selective. It's got spelling mistakes, I see, but it's got clusters at different sequence identities. And so you're evenly sampling sequence space, and that allows for faster searching. Now the problem, this notion of redundancy can be a bit surprising sometimes. Here's the result of me the other day just searching for human cytochrome B. It seems I got 1,964 cytochrome B entries in Uniprot for human. This is because it's mitochondrial. People who just used to put mitochondrial sequences for humans in willy-nilly, and yeah, there might be a SNP occasionally, but really there only ought to be one entry. So sometimes you can get overwhelmed with redundancy. This is a particular problem in human, of course, and an even bigger problem with mitochondrial stuff. The solution here is just to make sure you select only the things that have been reviewed, and that's usually an easy check to make. Okay, so there's lots of protein sequence stuff. Of course, we want to group these things together into families. There's 3 websites I commonly look at for this. TreeFam is quite pretty. Panther is very comprehensive. It's updated regularly. And very interestingly, it's taken a big effort to bring in lots of annotations to do with possible function or known function from the gene ontology, from Go. So that's behind the big comprehensive one. I would, if you sort of want details, TreeFam perhaps is my recommendation if you want pretty, but also it's also worth a look. As I say, these are just ones I've used. So these are groupings of proteins into families that mostly relate to orthologs. And so by orthologs, I mean similar homologous proteins that have arisen from speciation, so different versions of the same protein in different species. Of course, that breaks down the sort of more sort of evolutionary difference you have between your species. But these guys have in these databases good effort to try and make them relevant. And Panther actually has different levels of clustering, if you like. It has sort of local families and larger families, and so you can actually do some tuning there. Also be aware of this term called paralog, which is just a duplication event that means that there's a similar protein. And of course, this sort of muddies the water. Sometimes it's relevant to include close paralogs. Sometimes the paralog isn't what you really wanted. So you've got to be a little bit careful about that sort of thing. But there's huge amounts of data there. Now, when you're talking about families and evolution, at a protein, or most of this is in terms of the domains. The units of evolution are these sort of functional regions. So there's lots of data relating to that. If you want sort of bioinformatics kind of things like HMMs and sequence clustering, and then PFAM's the one you want. So if I want to annotate sequences using my sort of Python programming pipeline, it's PFAM, probably PFAM section that I would use. But there's also Interpro and ProSite, and they offer good user interfaces, and I particularly like ProSite because not only does it give you sort of the domain organization of of all the proteins that it has in the database, it also has a little tool where you can actually create your own domain style diagram to put in your slides and whatnot. That's very. You just type in, it starts, this bit called, say, LMB starts here, ends here, and is colored, whatever. It's very simple to use. Okay, and sort of getting more complicated. Of course, there's 3D structures we have to talk about. There's a notion of sort of 3D structures together into families, not necessarily just due to homology, but also due to 3D fold similarity. And so there's Kath and Scott, which of course is run by Alexi and others at the LMB. And the idea is they've got a sort of hierarchy of, these are close homologs, these are more distant homologs, and these ones probably have the same structure, but then I don't know about the homology being close. And so, if you're interested in actually looking at things on a structural basis, that's your start point in terms of secondary structures and how they fold. If you want the actual coordinates, well, there's only one place to go there, really, and that's the PDB. Although the PDB, as we know it, is officially called the WWPDB, which is a collection of actually 4 different groups. There's the main one, if you like, or the original one in America, the RCSB one, and that's probably where I'd go if I wanted to download the authoritative information for protein coordinates. There's the PDBJ Japan, and there's the E in Europe. Now, the PDB-E in Europe actually has a few tools there to do with protein structure, so doing alignments of proteins based on sequence and structure. You can do that there, for example. But it has other things it's developed, like PISA, and I'm going to have to look into this because I'm interested in looking at interfaces, and PISA has pre-calculated information to do with all the complexes and how proteins stick together. So that can potentially be very handy. Most of this, of course, will be crystallographic data, but there's one of the partners in the PDB is the Biomag ResBank, which is NMR data. So if you want not just coordinate, but you want the actual NMR chemical shifts and NOE restraints, that will be provided in there. And these guys are doing much better at sort of coordinating than many databases. So it'll all be consistent. Okay, onto function. Actually, there's an overwhelming amount of databases I can point you at here, but I guess the most important one is the gene ontology. And this splits annotations of what proteins do into 3 sections. First is what the molecular function is, how the enzyme works or whatever. The biological process, so this is in terms of why it's useful to the biological system or the species, and the cellular component is like where it is. And it's a hierarchical ontology, in other words, that is supposed to explain classes and then superclasses, subclasses of information. But be aware that the actual assignment of Go information can be a little bit ad hoc, and particularly it depends on what people have done, how it's been added. There's a lot more automation these days, but some of the old annotations have been stuck on, well, sometimes they can actually be wrong, but more importantly, more commonly, the problem is that the way that they're specified has been patchy, at least in the past. For example, I was looking at membrane proteins, and I was collecting things that were in the plasma membrane, so the outer membrane of human cells. Sometimes it says, this is a plasma membrane protein. Great, so that's the term they use. Sometimes the annotation will be, it's in the plasma membrane, and there'll be a separate one that it's a membrane protein. You have to combine the two, and sometimes it will be like apical, neuron membrane or something. You could be really specific. So there can be lots of terms that effectively mean the same thing with regard to what you're interested in. So my advice here is trying to go to the simplest, the highest level description, if you can. And also many things will do gene ontology enrichment, which has taken care of all that kind of nonsense for you. If you want to know what a gene or a protein does, actually an often forgotten kind of database are things like, which is online Mendelian inheritance in man, and that's kind of a literature collection, in this case of human genetic conditions. But to know what something does, it's interesting to know what happens when it goes wrong. I mean, biologists know that for a long time, and this kind of database stores that that information. Sorry, I missed the URL. Okay, so that's kind of the functions we know. Of course, all this is connected into pathways and their interactions. So there are some databases I've mentioned there. I like to use KEGG if I'm interested in, for example, just what's in glycolysis, kind of traditional biochemical pathways, or it does more than that. But if you want the kind of latest fancy graphics, it's a really nice website at Reactome. Actually, I was very surprised. So this is was it something to do with COP1 and 2 transport between the Golgi and the ER. Okay, that's a pathway that they describe. And I just went in, and they had already a pre-created cartoon-like diagram for that process. And they'll do every single pathway you can imagine in many species. And so if you want a slick website and nice graphics you can use, then Reactome is the one that I recommend at the moment. Interactions, okay, again, lots of databases to choose from, so I've sort of pared it down to a couple of really nice ones. You can see the EBI databases, but check out the BioGrid, that's up and coming, obviously. And I'm actually involved in creating a database of my own things. So this is with Sean Munro and Matthew Freeman, who's now in Oxford. And what we did is we actually looked at Go annotations and the idea is that the amount you know about a protein can be assessed from how much annotation it has. What is its knownness? So Sean and Matthew thought of the idea of score, and here's a plot I made where you plot you sort of plot knownness of proteins or their families. We treat things as families, so that, you know, if you've got a molecular organism where lots of experiments have been done, that information spreads out to the other things in the family. And this amount of information is concentrated in a relatively small fraction of the genome, and sort of 10 to 15%. This is shrinking, of course, where we know almost nothing. And the annotations that get splattered on tend to be automated, not things that humans at log scale, by the way. You know, there are some things we know an awful lot about, but most we know a tiny bit. And so to basically quantify that and allow you to sort of search through, you know, which things don't we know about so much. And she hosted at the LMB. I'm not sure it's gone active yet, but it will be in the next couple of months if it's not already. Of the most known protein in the database is beta-catenin. Okay, and this is one that's very unknown. It has one automatic annotation, but it is, the things we've put in the database tend to be things that are in lots of species. So this is very well conserved, important, but it has no annotation. So, you know, maybe this can give you some ideas for where to research next. Okay, now on to tools. I see I've got a question. I'll take that at the end. Keep the questions coming, of course. Okay, so databases is where you get your data, is doing something with them. Again, it's a light overview. Please tell me if there's something you think I should put in this talk for next time, because these are just my personal, for things that you can mostly get on the web. Some programs can be installed locally, especially if you want to pipeline, of course, that's important. So this is a light overview. I'll go through some specifics, but as before, if you want general stuff, go to the hubs. So the Tübingen one, there's one at SY and the EBI, you know, loads of tools in there. You can just run them straight off the website. You know, that's where I go to start with if I don't know what I'm doing. Okay, so alignment tools. So if you want to do really big alignments, well, my recommendation is try to. There are programs like LastC, MultiZ, which does multiple LastC genome alignments, but genome alignment anyway, it takes a long time. You've got to really know what you're doing, particularly if you've got lots of species. But fortunately, there's a lot of data of pre-computed stuff out there. If you want to, you know, alignments of primates, it's been done. You know, you want alignments, you know, with all the organisms, you know, it's been done. So, so go there to actually get the data. Don't try and do it yourself. Interestingly, if you're for aligning small regions, I'm a big fan of the really old program called ClustalW. And the reason for this is that it's gapped, you know, inserts gaps, which some of the short read aligners over here don't use, but it's also very quick and uses very little memory. Okay, so you know, despite being very old these days, I still really like to use that one. And there's lots of aligners you can actually look at this website here. That's ClustalO, not ClustalW. I put. Sort of another thing you can do with the DNA sequences is you can actually align to proteins rather than just do DNA alignments. And a reason to do this, for example, will be to find where a protein annotation is missing. So you can take a protein sequence and you think, well, this ought to be represented in the genome because it's conserved. And you can actually look at using, for example, tBLAST-N, at the translation of the genome and how that matches your protein. And that you can find, you know, fragments of proteins and see why things have been misannotated. It's really good for, you know, for that sort of thing. And if you're using protein sequences, then they will actually have a higher signal to noise ratio because they're slower to evolve, and you can link protein sequences according to similarity. And there's only 4 bases, but there are 20 amino acid residues, and they have biophysical properties. So protein sequence, if it's relevant, is the thing you should do, rather than just DNA alignment, of course, for the open reading frames. Most alignment these days, of course, is to do with the kind of high throughput sequencing technology that mostly Illumina provides. And so the aligners, actually I use most, are things like Bowtie and BWA. You can get a billion reads out of an NovaSeq lane these days. Okay, so these are deliberately designed to be fast, fairly memory efficient. I'm not going to talk an awful lot about them, but here's some starters. These 3, you pick and choose. There's subtleties that are different between them according to exact applications, but they're all very good. They're all very fast. I tend to like Bowtie 2 the best because I know how to mess with its parameters. Okay. Note that if you're discovering SNPs or you want to do ChIP-seq, these are the aligners to use. And many pipelines that you will find will have these built in. But be aware that if you're doing RNA-seq alignment, then actually different tools are required. So like STAR or Salmon. And the reason is because RNAs are spliced. You can't just, the read might have to jump over an intron. So a different kind of technology is required there, or you could get exome sequences and all sorts of things. So just know that the RNA splicing uses its own set of software. But I won't be going into RNA-seq in great detail. There just isn't the time. Okay, so all this short read information, you can use it to find mutations, and of course do sort of biochemistry like this to find where, for example, histone modifications are. But to actually start to do this, to visualize and whatnot, we need some sort of workflow. Huge number of ways of doing this, depending on exactly what your biological question is. But the general process will be that the machine, the sequencing machine or the service, will call the bases for you. So you'll get GCA and T and N's, unfortunately, back. And those raw reads with quality scores are then what you actually have to deal with yourself. Always do QC on your base sequencing stuff. I recommend FastQC by Simon, who's at the Babe Room. Also, be really aware that you'll have adapters in there. These things need chopping off. There'll be, you'll have got little bits of DNA out, however, and you'll have all of your synthetic sequences stuck on the end. So you need to assess that it's good quality natural sequence. And then there's various ways that you'll align, as I've mentioned, to the genome, and then do various analyses. And again, there's so much detail in there, I can't really go into it, but a tip is to try the Sync application if you want to analyze data. It's actually a desktop application. You download it and install it, so it's perhaps a bit old-fashioned in that regard, but it's really easy to use, and it's quick, and it's really quick at answering questions. I like it very much. So if you just want to put on your ChIP-seq or your RNA-seq onto the chromosomes, have a look in a very quick manner and not be dependent on websites, that's the one to use. Now, when you attempt to think about a pipeline that would be useful for your experiments in terms of the information you get out, then there's a few pointers here I'd like to draw your attention to. One is, how much read depth do I need? Okay, so depth means how many times is each base pair position represented? And the obvious thing is that the more depth you have, the more signal more sensitive you are. So the more you can cope with backgrounds and things. But of course, this question, how much you need, depends on what you're trying to do. Okay, so if you're just a light touch to look at one or two specific aspects, then you might not need a lot of depth. A lot of this actually boils down to how should I multiplex my data? So that means how many barcodes do I want? How many different samples am I going to barcode and sequence at the same time? If I want more depth, use fewer barcodes. It's as simple as that. But if you're doing an RNA-seq run that's just to check an easy differential expression, you might be able to run sort of more than 20 samples on one less powerful sequencing machine. Although if I want a high C experiment, which is chromatin folding and stuff, I might want actually all of the billion reads from one lane of a NovaSeq. So just be aware of that kind of compromise. Okay, so actually doing the sequence processing, once you've got the reads, there's a vast amount you can do. Here's a few pointers for things that you can do mostly on the command line, and also using interfaces like Galaxy, for example, where it's sort of a graphical web interface. You can chain these things together. And I hasten to add, at Babram, they have sort of pipelines. They stick together themselves, and at the LMB, we're also beginning to stick together our own pipelines, and also we try and make them available in the future on the web. So hopefully look forward to seeing some of those announced. Anyhow, to start with, make sure you take care of all the trimming of adapters, of quality scores. Have a look at FastQC and TrimGalore. The Babram guys have done a great amount of work there. SAMTools and Picard are very common if you want to start looking at aligned reads. So where it knows where there is in the genome, and if you want to start sort of piling them up and all sorts of things. If you're interested in sort of messing around with dating them, reformatting them, all sorts of things, bed tools, and there's also HTC things that I like to look at. For there, a couple of key players. So this is if you want to look at mutations, sort of call where your insertions, deletions, etc. are. GATK is very comprehensive, got a lot of documentation. It's very established. I find it can be a little bit picky with the formats, but in a way that just means fewer errors, I guess. I'm also a big fan of Freebase. This uses sort of more slightly more intelligent response method to actually calling SNPs, etc. And it's very quick actually, but it's always a bonus. And the idea is with these that you can pile up they pile up your DNA sequence reads, and they look for differences to the reference genome. Of course, they've already been aligned at this stage, but also highlight where you've got changes, insertions, deletions, and particularly Freebase is able to call different haplotypes that may have been sequenced at the same time. So you get a whole bunch of people with slightly different versions. It will give you with probabilities the different mutations it thinks exist. Though they're fine to use if you just want to work on one kind of sample. For protein sequence alignment, I guess there's two sort of ways of doing this. There's just the quick way to find proteins in the first place, and there's the alignment of multiple proteins that are related to one another, multiple alignment. Now, most people know and have used BLAST. That still remains a good option, it's still quick for searching for protein sequences that are likely homologous. Be aware, of course, it is not the most sensitive method out there. And of course, what's happened these days, computers are getting faster and faster. So the more sensitive methods that require a bit more compute power, you wouldn't notice any, speed difference particularly. There's SciBlast, which if you just want a fairly sort of fast and easy way into more sensitive alignments, you can just run instead of regular BLAST. What it does is it builds a profile, an alignment, and then searches with that again. And that's more sensitive. There's HMM-based methods which give even more sensitivity. Hummer, which I like very much, and there's an iterative version. So it's building an alignment, improving alignment as it goes along to get more of an idea of what's in your protein family, and it researches. And the ultimate is the profile alignments, and the HH suite does that. And that's basically you're sort of comparing two clusters, much better than comparing two sequences or a sequence with a cluster or family, because you actually a lot more information and you can detect more remote homology. In all of this, you tend to get an E value, just to things typically say how significant a hit is. This is the number of hits at this score level. That's what it means. So if the score is 10, I'd get 10 at random. Well, then my hit is probably not going to mean very much. So it was much less than 1. 10 to the minus 3 is borderline. 10 to the minus 10, that's really good. But beware with e-values that and also the size of the protein. The smaller the protein, the more easy it is to match the sequence with other random sequences. And so actually, if you're searching for sort of 30, 40, 50 amino acid region, the e-values and the typical cutoffs can be unhelpful. You have to be more relaxed and not worry if your e-value is 1. Okay, you pull out the hits and then you go in through other means and say, well, actually, is this meaningful? For multiple alignment, use the hub sites. I list a couple here, and the idea is, of course, that you make a pro sequences, and you can do that. You can use that to look at conservation. Here's some pretty stuff I'm doing on our website. It's going to appear at LMB at some stage. I really like muscle. That's been tuned to use different strategies if it thinks something is the folded hydrophobic part, and also cluster omega. That's my workhorse. That's my current recommendation. And I like the fact you can align to an existing. You want to add another sequence, you can use cluster omega. That's really handy. And this uses all the latest HMM-based maths behind it. And actually, when doing these sorts of things, I figured I really realized it's the same strategy. So I'm going to share that with you now. It's a bit like what SciBlast does, which is where you grow an alignment that if you search initially in a smallest database as possible, get few relevant hits, maybe relevant species. And then you filter your alignment, so you can remove anything that has too many gaps, you can chop off the edges of the alignment, and then you research. And then that will focus your searches on only the things you're interested in. Okay, and for example, if your protein has a kinase domain, you might find there's all hits come from every kinase known to to mankind. But focusing this will make sure that you can throw away at an early stage those sorts of aspects you don't want to search into. So typically I will take the best hit in just a limited number of species and alignment on that if it looks like they're genuine orthologs. So that's a strategy that I recommend. Phylogeny. Oh, there's a great workflow for phylogeny at phylogeny.france.fr. It will do the multiple alignment for you, filter the alignments, construct trees. If you want simple stuff, that's what I recommend. If you want to do it yourself, there are various kind of ever increasingly complicated methods of tree building. The ultimate is Mr. Bayes. Properly mathematical, you can all sorts of probability and modeling, Markov chain stuff in there. So if you're really into building trees, Mr. Bayes is your man, I guess. For drawing pretty pictures like this, I recommend Dendroscope. It's sort of an installable program, but it's very easy to have all sorts of different kinds of pretty trees. Okay, how are we doing time-wise? Okay, I'm going to speed up a bit because time is running a little short. So here's the basic pipeline for comparative modeling. I point you in lots of tools here. Swiss model does everything for you, but you might want to do separate steps yourself. Modeler is great for actually making the models. Now, creating models using homologs, comparative modeling, is only good if the homologs are close, but actually it's probably the best method if you've got a really close homolog, because you're not changing very much in a way. There are things like TR Rosetta and EV couplings where actually it will try and fold any sequence for you, and those are online, so give those a go. TR Rosetta has been improved. Rosetta's been improved with deep neural networks, and it came second in the CAS competition. EV couplings using this idea of coevolution, providing distance restraints to fold the proteins. So you can actually solve fold protein structures online. Okay, enough with tools. You want to do your own stuff? Get into programming, learn a bit of Python first, then you can go on to R or maybe some of the compiled languages. You can actually dabble online with Google Colab if you want to try a bit of Python. Hopefully you'll put all your stuff on GitHub when you've made something nice. Please share. Also, you have to learn a little bit about the command line. We run courses here. There's courses available in the university if you want to actually start programming, and of course, loads online. I won't go through this, but here's a list of some file formats. You might want to know what they do before you worry too much. So these file formats underpin lots of the tools that I've been talking about today. Oh, excuse me. So check that out. And actually, there's a helpful, if you're into sort of genomics or epigenetics, etc., UCSC has a really handy description of what exactly a big file, whatever it is. So there's a lot of stuff explained in here about actually how the formats work. Also, sort of as a sort of compendium for you, I've compiled a list of statistics pointers, which I feel would be helpful. A lot of questions I get as a bioinformatician are, how do I check that this is significant? What test do I use? What's the difference between two ratios? Are these ratios? So I've tried to compile a little list here of stuff like that. And the thou shalt not forget. Know whether your data is normal. Have you tested for multiple testing correction stuff? Do I need to use an exact test? These are things you have to go in and understand yourself or ask an expert about. And here are some recommendations. There's quite a lot of which test do I use? Are two ratios the same? Well, Fisher's exact test does that. Okay, and it's not phrased like that when you read it on the Wikipedia article. That's what you can use it for. So there's lots of data there. I'll make sure these slides are available if you want to use this as a resource. Final sort of tips for if you're going to go into the programming is please remember to try and do controls. People do wet lab controls, but you need bioinformatics. Have positive and negative examples because bioinformatics pipelines can fail pretty silently sometimes. And of course, you don't just make predictions, try and test them out. That's obvious. Also, if in doubt, visualize. Go and have a look at your outliers. Have a look at a range of different things. Pick out the outliers. Go and see why do I understand why it's different. And here's one last sort of note on this is that sometimes it's not the bioinformatician's fault. Sometimes the data is just not usable for what it was intended. If you don't have the right data, you can't make the right kind of decisions. It was a surprise to have this correlation, but I did get to understand why in the end. Okay, so just to finish up, all of these kind of, when you get into bioinformatics, you're kind of steered towards this way of thinking about data, and it sort of eventually goes into this idea of machine learning. Something you might do to start with is clustering, just sort of plotting things, having a look. Here's some pointers on the method you can try. Top recommendation is please have a look at just the simple hierarchical clustering. It is quite easy, it's quite quick, and it gives you a good understanding quite often of how close things are. Sometimes these other methods, particularly if you're data multidimensional, can be a bit kind of misleading. So I'll leave you to have a look at that. But when you start clustering, you can't, and doing these sorts of things, you begin to visualize your data as points. And if you can describe all your data as a tensor, so that's an array of numbers in multiple dimensions, you can begin to start to use machine learning methods. So, if you've got a time signal or an image, that's just intensities. It's a list of intensities. If you've got a color image, it's just a list of intensities for red, green, and blue. So there's three, it's 3 deep. Even if it's just if it's biological sequence, you can make it into a vector or a tensor by, for example, putting a 1 for each position on the channel for G, C, A, or T. So you can think of this as sequences 4-channel data. And only one channel is selected at a time, but you've got 4 channels over the length of your sequence. So you can put data in as numbers, and if you can do that, you can begin to do many of these machine learning things. And in a way, all machine learning is doing is it's just trying to find a model, a pattern, by drawing boundaries effectively. So at a very simple level, actually, I've got a very simple example here. This is a very simple machine learning method called the k-nearest neighbors. There are lots of methods. Actually, here's a good collection here at the scikit-learn. And these don't require big computers to do it. You can just go in and have a play with that Python library. But all machine learning is in a way, is what this algorithm does, which is to have data that's labeled, okay? And when you have a query, just finds a number, k, nearest things, the things that are most similar. It's not obvious how you measure distance here. And then you poll. And so this may be closest to 3 class Bs than class A. So you say it is also class B. That is as simple as it gets in a way. You have a model and you predict from the model. And fancy neural networks in a way are kind of doing the same sort of thing, although obviously there's a lot more sophistication going on. And this is the ultimate, of course, of where you get to. This is AlphaFold from Google DeepMind. This is a neural network. It is huge. There are 220 residual blocks. It's too big to run on on even a big graphics card, I reckon. But you can win the CAS competition handsomely if you have enough compute power and neural network layers. So if you get into computing and you think you can describe things as vectors, there's loads of different methods that you can sort of avail yourself to. All right, I'll stop there because I'm a bit over time. I'll stop the share and we'll take some questions. George Souza says, I sometimes need to analyze prokaryotic genomes to check which genes are nearby specific target genes. Okay, first I go to KEGG to see if the genome is there, but it's quite an incomplete database. If the particular genome isn't available in KEGG, then I go to NCBI, which is not user-friendly, just lists contextual genes. Do you recommend a specific database for gene cluster analysis in prokaryotes? I don't do a lot of prokaryotic work, so I'll have to get back to you on that one. I know some people who do, who work particularly with the cyanobacteria. So my advice is there anything specific for your prokaryotes? Is there like a cyanobase or equivalent? Maybe check that out. Otherwise, I'll have to get back to you on that. Sorry, I'm too much of a eukaryotic guy at the moment, at least. But I will note that down. Okay, Caddy, what key methods would you recommend for comparing populations when data sets are not normally distributed? Okay, so in my slides, I would recommend one of those non-parametric tests. Go back and have a look at the exact name. So there's the Mann-Whitney U-test, and so that's for pairs. You're comparing pairs of, so 2 groups of data, nothing to do with paired. And there's the Crystal-Wallace test, which is effectively a multi-set version of that. And these work on ranks. Effectively, they're asking the question, are the medians the same? But like a t-test or whatever, they will give you a p-value. Okay, so check back on my slides or email me if you can't find it out. But yeah, non-parametric tests like Crystal-Wallace and the Mann-Whitney U, what I do. There's this other thing you can do if something's not normally distributed but can be made to be normal, for example, using a box plot transformation, then that might be better for you. I remember we were doing some studies of Drosophila wing areas, and we actually found that the thing we were measuring, which was the wing area, was the area itself was not the thing that was normally distributed. There was like a square root involved or whatever, because it was an area. And so if you have a power transform, basically just do something to the power of some, all of data to the power of some number, which you can optimize with the Box-Cox transform, then your data suddenly becomes much more normal, and you can do all the usual tests like ANOVA or t-tests or whatever. So that's my recommendation there. And Connie, do you have any recommendations on how to look at intrinsically disordered proteins where conventional alignments are not useful? So I guess identifying, yeah, I've done a little bit about this. So I actually once helped people, actually in Gene Thomas lab in Biochem, how to turn a disordered protein into an ordered one. It turned out that we could swap a loop from another protein and it suddenly became stable. I would have a look at small scale disorder. Look at things like sequence entropy, which is basically a measure of repetitiveness. I think you can plot these in with some of the tools at XPath-Y. If you can't, then maybe I can help you do that. Also, secondary structure prediction still works to tell you what the tendencies of the amino acids are. So, you know, particularly this disordered protein I worked on, it was only folded when it stuck to DNA. If you ran things like JPRED and these secondary structure predictors, then actually you could see it's predicting strongly helix here, strongly beta sheet there. And actually, when we did get it to fold, those were the beta sheets and alpha helix things. So you can still use protein secondary structure prediction tools. There will be disordered region tools as well, and sequence entropy is a simple version of that if you want to do it. Get back to me if that answer is not satisfactory. Okay, is that it, Stephen? Any other questions? Oh, there's more. More have come in. Okay.
Yeah, there's more come in. You've got the knowledge.
Okay, there's some more here. Do you have any recommendations for protein localization prediction software? For the Golgi. So actually, Sean and I worked on this. For membrane proteins, you can predict Golgi plasma membrane to about 70%. And actually, we've got a neural network, whether the website's working or not. So if you can't find it from I think it was a Cell publication a while back, then get in touch with myself and I'll point you at that. So that's okay for membrane proteins. There are other tools to actually predict any protein. Deep lock. So deep lock, I think, was the answer there, and sorry, is the program there, and it's a big neural network that takes all sorts of things and looks for nuclear localization, and it should be fairly good on the secretory pathway as well. And the other kind of data to look at is proteomic data that's spatially separated. There's a kind of experiment called LOPIT, which uses isobaric tags on basically organelles that have been spun down. And people usually represent this as a 2D map where they can sort of see all clusters of different organelles, and you can see Golgi ER. Now, if your data is in there's probably mouse data, there's also definitely lots of human data, there's also lots of plant data. Look at published LOPIT data sets. It's a little bit insensitive for the low protein concentration, but that is a very robust way of seeing which organelle your protein of interest is in, and can do Golgi discrimination. It is cell type specific, of course, but it'll be a good indicator of some of the major ones. Okay. George sent me a follow-up. Okay, well, please email me to remind me if you need more help on that. And one from Nina. More of a wide question approach proteomics level data? Are there any databases that offer additional detailed information on the physiological context in which the samples were extracted? Simple answer is I don't really know, but have a look at ProteomeDB. Extensive information on the original experiment in there. Probably also PaxDB. Mostly this will be due to tissue type, but I don't know these. I'm afraid. Just have a delve into the big databases. If it's cancer cell lines, then DepMap has proteomic information as well as the genomic stuff I'm mentioning as well. And so you'll have, hundreds of cancer cell lines of different developmental origin in there as well. And they're all, tell you exactly how everything is done. And that will be in a reproducible, single manner. Okay. Right, unless there's anything else. Okay. All right, I think there's nothing else, and I'll stop there. Do you want to say anything, Stephen?
No, just thanks for everybody staying on for the
all right. Well, see you all.
Questions, yeah. And we'll see you again on Thursday, then, when we'll continue on about with a bit of proteomics, but from a mass spec direction, where Mark's Keel will give a talk. The first of his 2 talks to finish the series. So please, everybody, back again. We'll see you on Thursday.
Yeah. Oh, and before I forget to mention it, I sort of headed into the realm of machine learning here. We're considering adding a machine learning talk to this series, but in the meantime, what I'll probably do is offer a machine learning lecture, just general intro, later on in the year. Okay, so stay tuned for announcements to do with that. It'll be nice and light touch, but like I did today, it'll hopefully get you started. Okay, take care, everybody.